In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Sperm develop in the seminiferous tubules of the testes. Primordial sperm cells, called spermatogonia, lie dormant in the outer compartment of the tubules from the late fetal period until puberty. At puberty, the spermatogonia undergo mitotic divisions, giving rise to genetically identical cells. The cells grow gradually into primary spermatocytes, preparing to divide into cells half their size. Each of these primary spermatocytes undergoes a first meiotic division, forming two smaller haploid cells known as secondary spermatocytes. These secondary spermatocytes divide once more, giving rise to four haploid cells called spermatids, containing exactly half of the father's genetic material. Through a process known as spermiogenesis, the spermatids change shape, transforming into mature sperm cells. The spermatid's nucleus condenses, the acrosome appears, and the tail begins to grow. Much of the cytoplasm is shed. As spermiogenesis completes, the sperm cells break free from the Sertoli cell lining of the tubules into the lumina. The sperm cells are then stored in the epididymis, where they functionally mature.
At puberty, a woman has about 400,000 primordial follicles, each of which contains a primary oocyte surrounded by a single layer of granulosa cells. After puberty, 5 to 12 primordial follicles develop into primary follicles each month. Only a few primary follicles continue to grow, developing a fluid-filled cavity called the antrum. Eventually, one becomes dominant. At the time of ovulation, the mature follicle moves close to the surface of the ovary. The fimbriae lifts off the expelled oocyte for potential fertilization.
So today we will be looking at the gametogenesis in plants and particularly in the flowering plants. So first of all, we will focus on the male gamete formation that takes place in the anther of plant. So to start with, we have a diploid pollen mother cell as you can see on your screen. This is the diploid cell that divides by meiosis. So we have our first meiotic division, and then there will be a second meiotic division. So we end up with having four daughter cells that at the end of meiosis. and they all are, of course, haploid cells. So let's consider each dots uh, as a haploid nucleus inside a cell, and each of these cells will divide by mitosis. Now, each cell will have two nuclei, as you can see, but the division is not completed because cytokinesis do not takes place at this point. So, we have one cell having two haploid nuclei, and that happens with each of these cells. So now one of these is called as tube nucleus, while the other one is called as generative nucleus. And these cells will then just become mature, and each of these cells becomes a pollen grain. So as you can see here, that one pollen grain contains two haploid nuclei. One of them is called as tube nucleus, while the other one is called as generative nucleus. So now let's talk about female gamete formation. The female gametes in flowering plants are formed slightly differently, but we start off again with the diploid mother cell. As you can see in your screen, this time we call it as the spore mother cell. And again, this spore mother cell will divide first of all by meiosis. So we have our first meiotic division, and then we have our second meiotic division. But the difference here, though, is that out of these four daughter cells that will end up, three of them will degenerate, or you can say will disappear. So it's only the one cell here which can continue on to become a mature female gamete. So now I'm going to replace this with a little dot to represent a haploid nucleus, and this is now known as embryo sac. Now this embryo sac gets bigger and it divides by mitosis. So we end up now with an embryo sac which is a much larger, and it has two haploid nuclei inside it. We then get a second mitotic division, which means we end up with having four haploid nuclei inside the embryo sac. And then we get one more. So we have our third mitotic division, which means we end up with having eight haploid nuclei inside. So three of them move towards the one end, while the other three of them will move towards the other end. But there are two nuclei which are present in the middle of the embryo sac. These are called polar nuclei. So these polar nuclei will then fuse together, and out of the haploid nuclei that are present at the ends, one of them, usually this one which is in the middle, will become a female gamete. And remember that this is the embryo sac which is present inside the ovule, in which the male gametes from the pollen grain will fuse with this female gamete, or you can say haploid nuclei, during fertilization. So this is all about the gametogenesis in flowering plants.